In this video, we are going to talk about proteins. Now, before we talk about how to synthesize or create a protein, I first want to remind us what actually creates these polymers. The polymer of a protein is made up of individual monomers called amino acids. Each amino acid consists of a consistent carbon backbone containing an amine group, a carboxyl group. And so this forms the carbon backbone of the ultimate polymer here. And ultimately, there are 20 different types of amino acids. The amino acid identity is determined by this third component of the monomer called an R group. So this is a variable. This is going to be different depending on which amino acid we have. In this case, this particular chemical structure indicates methionine. So this entire molecule here, the amine, the carboxyl, and this exact R group, means that this amino acid is called a methionine. Another example, we have an amine, a carboxyl, and a different molecule up here, a different R group. This is called phenylalanine. And so here we have methionine and phenylalanine, two of ultimately 20 different amino acids. Now, when we want to make a polymer, right, so the protein itself, also called a peptide, we need to join these two molecules, these two amino acids, together. And so what we do is we remove a water molecule. So H2O is removed by an enzyme. We're going to talk all about that process in what is called a dehydration reaction, because we are literally dehydrating these two molecules, we're removing water. Also is called a synthesis reaction, because we start with two smaller molecules and ultimately have a larger molecule. We have synthesized a larger protein from two individual amino acids. The resulting bond in between these two amino acids is called a peptide bond. Okay? So, in order to create a protein, we first need to essentially look at the recipe for a protein. Where are these recipes stored? Well, they are stored within the nucleus. So most of this video is going to be walking through this diagram here, which is collectively called protein synthesis. To give us a little bit of context here, this is a cell, a eukaryotic cell. Within the cell is a nucleus. The nucleus is what contains the entire genome of the organism, in this case a human. Okay, so the entire genome is stored within here. It is stored in humans on 23 different types of chromosomes. Usually when you see these molecules, they're kind of bound up into this nice little figure eight looking chromatid form. In reality, during the normal day-to-day life of a cell, the DNA is actually kind of unwound and just kind of distributed throughout the nucleus. And this is important because we need to be able to read these recipes, which are stored within the genome. Okay? So this kind of cotton candy looking configuration of the DNA is called chromatin. Okay? There are particular genes that are stored within a densely packed region of DNA within the nucleus, which is called the nucleolus. Some of these genes include the rRNA genes. Okay? So these are going to be really important in creating what is called a ribosome, which is responsible for actually catalyzing reactions in between the amino acids. Okay. A couple more notes before we move into the larger picture here. These structures are called nuclear pores. Note that they are very specialized proteins which pass directly through not one but both membranes surrounding the nucleus. Okay, so we have two phospholipid bilayers surrounding the nucleus, making sure that this cookbook, this genetic code, is always protected. Now, these nuclear pores do allow things to pass from outside the nucleus inside the nucleus and vice versa, but it is very selective about what can actually pass. Okay? One of the main concepts of this story is that the DNA is way too big to fit through these nuclear pores. We need to maintain and we need to protect it within the nucleus, and so the entire cookbook cannot pass out into the cytoplasm or this outer region here, the cell. Instead, the cookbook stays in here, and we copy down a single recipe from the entire cookbook, and that's called mRNA, We allow that recipe to go out into the cytoplasm, and there we read it and make the protein. Okay, so let's start by talking about making a copy, making a transcript of that recipe. Again, we are interested in making one single type of protein. The code, the genetic information about this particular protein is stored within the DNA, within a particular region of the DNA called a gene. Okay? Now, if this particular cell is being told to make this particular protein, right, so the more videos on that at another point, essentially those signaling molecules can activate a region of the DNA called the promoter region. Okay, usually there are different types of proteins and other regulatory molecules that are attached to more or less the beginning of a gene. And signaling molecules can activate that or remove it as a block or something so that the rest of the recipe can be read, the rest of this gene. Okay? When this cell is essentially told to make a protein, make a particular protein, the promoter region, right, is activated or freed up in some kind of way, in a lot of different fashions, and an enzyme called RNA polymerase is able to access the DNA and start reading it. Right? So when it's reading it, what's actually happening here is it is creating a copy of the gene. Okay? In another video, I explain this process of transcription. Right, so here we can see the DNA. We know that the DNA is a sequence of nucleotides, here just signified by the individual letters. Okay? RNA polymerase recognizes that cytosine is complementary to guanine, and it's going to catalyze a reaction between these nucleotides. Okay? So ultimately, the RNA polymerase is going to create a complementary copy, right? A goes with U, T goes with A goes with U, etc., of just this particular gene in the DNA. Again, if you need a review of this, you can click the link above. Okay, so that is what's happening here in the nucleus. Okay? So here, this red line here is signifying an mRNA copy of the gene in the DNA. Remember that as nucleotides, as nucleic acids, there's always the beginning, the 5' end, and the end, essentially, the 3' end. Now this mRNA is not exactly ready to go out into the cytoplasm and complete this process. First, we need to remove the regions of the mRNA that don't actually code for a protein. These regions are called introns. Okay, so here is an intron and here is an intron. These regions of the mRNA actually contain the recipe. They actually contain the information needed to create a specific protein. So enzymes are going to come along, they are going to cut out these introns, cut out this intron, and essentially glue together the remaining exons. Now I know this seems a little bit backwards, it seems like the exons should exit and the introns should stay in, but it's exactly opposite of what you would expect. Okay, so we have left only the exons. 2 more steps before this is finally ready to go out into the rest of the cell. Okay? We add a cap, a particular molecule, to the 5' at the beginning of the molecule, which essentially is going to flag down structures out within the cytoplasm and say, hey, this is the beginning of the recipe. Okay? Also, we add a lot of adenines, right? So lots of adenines, called a poly-A or polyadenine tail, to the end of the molecule. Essentially, out here in the cytoplasm, the molecule is going to be degraded from the 3' end. And so, adding a bunch of essentially garbage mRNA to the end is going to give this molecule a little bit more longevity so we can make more proteins from it. Okay, so now, finally, after transcription, okay, making a transcript, making a copy, processing that mRNA, okay, adding a cap and a tail, this molecule is now able to exit through the nuclear pore. Okay? Remember that the nuclear pores are very selective about what can come into or go out of the nucleus, and so the mRNA is short enough to pass through this through this structure. Okay? Once it is in the cytoplasm, it will encounter what are called free ribosomes. These ribosomes are not membrane bound. They are simply composed of different types of enzymes as well as rRNA. Okay, this smaller portion, called the small ribosomal subunit, is going to start at the 5' end and essentially slide down this mRNA strand looking for something in particular. Okay? This is really important. Let's zoom in on it a little bit more. Okay? What I have here is the mRNA. Okay? Remember that mRNA is always read from the 5' end as opposed to the 3' end. Also know that even though we talk about the sequence of this particular nucleic acid in terms of the nitrogenous bases, in reality, each nucleotide consists of a phosphate, a 5-carbon sugar containing an OH on the second carbon, right, because it's a ribonucleic acid, and the nitrogenous base. Okay, so when I draw this red line here, what I'm actually showing you is all of this just reduced so that we can focus specifically on these particular nucleotides. Okay? So again, the small ribosomal subunit is going to attach at the 5' end of the mRNA molecule, and it's going to slide along the mRNA looking for a particular sequence of nucleotides. Now, it is reading this strand, this genetic code, in 3-letter groups. Right? Every 3 letters is referred to as a codon. Okay, so again, if we're using this recipe analogy, this is the entire recipe. This is an individual word. All right, so this is calling for flour. This is calling for sugar. This is calling for eggs. Okay, one particular codon, 3-letter nucleotide sequence, is going to always signal the small ribosomal subunit that this is the beginning of protein synthesis. Okay, so this is the beginning of a process called translation. Okay, so this start codon is always AUG, adenine uracil guanine. Okay, and once the small ribosomal subunit locates this start codon, what is next going to happen, if we come back over here, is the large ribosomal subunit is essentially going to sandwich the mRNA in between the large and the small subunits. Okay, next, this structure right here is going to be called based on this 3-letter codon sequence. All right, so let's zoom in on that a little bit more again. Okay, note that AUG is complementary to UAC. Okay, now what is this funny looking molecule up here? Okay, this is called a tRNA. Okay, as an RNA, it is a single-stranded molecule, right, made up of A's, G's, C's, and U's for uracil. Okay, even though it is a single-stranded molecule, there are complementary nitrogenous bases throughout the single strand. Okay, these complementary bases will automatically, naturally form hydrogen bonds in between them and essentially fold this single strand back on itself. Okay, so what we can see here is a UG on this part of the tRNA strand, and AC on this part of the tRNA strand. In the cytoplasm, they are going to fold on themselves to essentially produce this, if you use your imagination, this T-shaped molecule. That is not why this is called tRNA. In fact, this is called transfer RNA. So why is it called transfer RNA? Well, this molecule is going to transfer a particular amino acid, in this case, phenylalanine. Okay, so remember, phenylalanine consists of amine, carboxyl, and this particular R group. Okay, so this entire thing right here is generally just portrayed as PHE. Okay, so this is the entire amino acid. Okay, and this this tRNA is going to transfer the amino acid into the ribosome. Okay, how does it know which amino acid to bring? With the anticodon. Okay, so this is a particular part of the strand of tRNA. Okay, AAG is complementary, we go back down here, to UUC. So this anticodon is going to form hydrogen bonds with UUC, the next codon, the next instruction for ingredient, right? The word within the recipe. Okay, so these 3 nucleotides form hydrogen bonds with the 3 within the codon of the mRNA, and this particular tRNA is going to bring phenylalanine. Okay, so at this point, we have had a tRNA bring methionine in response to the AUG start codon. Also within the ribosome, we have another tRNA bringing phenylalanine because of UUC, the codon within the mRNA. And now inside the ribosome, there are enzymes that are going to catalyze this reaction. Okay? Enzymes pull hydrogen, oxygen out of these 2 adjacent molecules and form, right, catalyze the reaction to form a bond, a peptide bond between methionine and phenylalanine. Now, how we portray this is simply this little circle here, methionine, and a line, and phenylalanine. So now we have this 2 amino acid long protein. And of course, this is not the end of the story. It's far from the end of the story. The entire ribosome is going to keep sliding down this mRNA, bringing tRNA molecules complementary to the next mRNA molecules and bringing the appropriate ingredient along, so the appropriate amino acid. Now, how do we know which amino acid to bring? Well, in many textbooks, there are charts, such as this chart right here. What you can see is a list of each and every possible codon within the mRNA and which amino acid is going to be brought based on these particular codons. Okay? Note that there are 3 codons that indicate that this is the end of the protein. Okay, so only 3. Also, AUG is always the start, okay, even though it also codes for methionine. Okay, so let us go back down to our ribosome here. Okay. Again, we have transcribed an mRNA copy of the DNA up here, processed it. That transcription, that copy, exited out of the nucleus into the cytoplasm. Okay. In the cytoplasm, the ribosome essentially slid along the mRNA looking for the start codon. The appropriate tRNA brought its amino acid. Okay. The next tRNA brought its amino acid. A peptide bond was catalyzed in between the 2 of them. Okay. This tRNA essentially leaves, gets lost, and the entire ribosome is going to slide down the mRNA, synthesizing this protein. Right? And again, this is with synthesis reactions. Now, many proteins aren't simply a simple strand of amino acids. They're a little bit more complicated than that. And so within this sequence is going to be a signal to essentially take this entire complex, ribosome, tRNAs, growing protein, mRNA, this entire complex is going to essentially dock on specialized proteins within the membrane of the rough endoplasmic reticulum. Note that the RER is simply an outpocketing, a specialized organelle connected to the outer layer of the nuclear envelope. Okay, in the RER, what we have is the growing protein is going to be directly inserted into the RER. Okay, so note that out here, the ribosome is still proceeding down the mRNA. Okay, it is still catalyzing these reactions. And the growing chain of amino acids, right, each one of these little green dots here is an amino acid. The growing protein is going to enter into the rough endoplasmic reticulum. Now, in the RER, there are a lot of enzymes that are going to process this protein. What does that entail? Well, each protein is different, so each one requires something a little bit different, but in general, what can happen is that sometimes this entire sequence, this entire chain of amino acids isn't necessary to the final functional protein. Instead, some of those amino acids are sometimes trimmed off. Okay? Also, the protein is generally functional in a secondary and tertiary structure. So the secondary structure is essentially forming hydrogen bonds between different amino acids. Tertiary structure is folding the entire protein into a three-dimensional functional shape. Okay, again, depending on the type of protein, this process is going to change quite a lot. Okay, also, some proteins function with associated carbohydrates. Right? So I like to think of this as kind of planting carbohydrate little hedges. Here we can see a globular protein, lots of hydrogen bonds in between, lots of alpha helices and beta pleated sheets, and these little fluffy things sticking off the outsides are extra carbohydrates. Again, this is very much dependent on what the protein actually is. Okay? Now, this process, of course, is not finished. This protein has a couple more stops to make before it is actually functional. Here, we can see that the protein is kind of pushed to the side within the rough endoplasmic reticulum. This phospholipid bilayer is going to be pulled inward, essentially creating a little droplet, I call it a transport vesicle, containing this protein. Now the next part of the story, the next step after the rough endoplasmic reticulum is all the way on the other side of the cell. Note this is the edge of the cell. Here is outside the cell, so the extracellular space, also called interstitial fluid. All of what we've been looking at so far is the cytoplasm or cytosol, right? So outside the nucleus, but still within the cell. Okay. The protein needs to be carried to the next organelle in line, which is called the Golgi apparatus, sometimes called the Golgi body. Now, in order to actually transport this little vesicle across the cell, we use one of my favorite little types of protein. This is called a molecular motor. There are different molecular motors that essentially act like trains to go away from the nucleus or towards the nucleus. They use a single ATP to move these little like Mickey Mouse-like legs, and essentially they're going to cart this transport vesicle along these cytoskeleton fibers towards the Golgi apparatus. Okay, so they use ATP to march along this cytoskeleton fiber and finally deliver it to the Golgi apparatus or the Golgi body. Now, because both the RER and the Golgi apparatus, and frankly, the plasma membrane of the cell, as well as the nucleus itself, they are all made of a phospholipid bilayer or surrounded by a phospholipid bilayer. So the membrane of the RER can simply fuse with the membrane of the Golgi apparatus, releasing its contents inside the first of about 3 to 20 independent little saccules. Okay? These are within these saccules, the protein is going to be processed a little bit more. Okay, so again, depending on what type of protein this is going to determine what processing is done. Some options include trimming these little carbohydrate hedges, right, making them exactly right for a fully functional proteoglycan, for example. Also, in some cases, even more amino acids need to be trimmed off, right? So the carbohydrates and or the protein can be trimmed within the Golgi. Now, the protein is passed from little saccule to saccule, again via these vesicles. So essentially pushing the partially processed protein against the phospholipid bilayer and creating a little droplet. Okay. Other processing that can occur here is creating the quaternary structure. Okay. So sometimes the proteins only function in association with other proteins. For example, hemoglobin is actually 4 different independent proteins joined together. And the only way that hemoglobin works is if all 4 of them are present and in their correct primary, secondary, tertiary, and quaternary structure. Okay. Another example, here we have a receptor that is coupled with a G protein. Okay, so this association between these proteins is critical to the function of the receptor. And so these molecules are going to be joined together in their appropriate way within the Golgi. Okay. Finally, once the protein is totally processed, it needs to be sent to its final destination. Now there's a lot of different options, right, because there's a lot of different proteins. I am going to show you 3. Here, we can see the finalized protein is packaged into a vesicle, right, just like we have seen so many times before. Okay. This vesicle is transported again by little molecular motors to the surface of the cell. Now, once at the surface of the cell, a lot of times these vesicles are just going to kind of hang out until they are triggered to fuse with the phospholipid bilayer. And again, this is a phospholipid bilayer. When I show you this little line here, I'm essentially indicating this. Okay? So when the cell is triggered, this vesicle membrane is going to fuse with the same type of structure, right, the phospholipid bilayer here, and open, dumping its contents out of the cell. This process is called exocytosis, and it's really important in releasing hormones, right, releasing neurotransmitters, releasing other types of secretory products. Okay, another option is inserting membrane proteins within the membrane of the cell. Here we have a protein that functions as a channel. Okay. Here we have a receptor protein, in fact a G protein coupled receptor. Okay. These are embedded within the membrane of the Golgi apparatus. Okay. A vesicle is formed, transported to the surface of the cell, fuses with the surface of the cell, and essentially this vesicle membrane is now part of the cell's membrane itself. Okay, so here we can see the channel protein, here we can see the receptor protein, and ultimately now the channel and the receptor are part of the membrane. And so this is essentially how different types of ion channels, for example, things like the sodium-potassium pump, right, different types of receptors such as the G protein coupled receptor that we are looking at here, all of these are ultimately inserted in the plasma membrane by, all the way back here, turning on a gene for the sodium-potassium pump, for example, transcribing the gene, processing the mRNA, translating the mRNA, processing it in the RER, transporting that vesicle and the protein across the cell, finalizing this protein, joining it together with others, and ultimately inserting it into the plasma membrane. Okay, so a lot of times we can change for example, permeability of a cell to different types of ions by inserting more of these channels, and we have to go through this entire process in order to get that protein. Okay, final example of destinations for this protein that we just spent so long making. Again, we can create a vesicle off of the Golgi apparatus. Okay, and this time the vesicle is going to become its own type of organelle. Okay, so here we can see a lysosome. These are different types of enzymes that were again created through this process. Okay, within the lysosome, right, so essentially like a circulating stomach throughout an animal cell, the pH is really low and it has lots of digestive enzymes. So again, really similar to a circulating stomach. Okay. So what does this do? Lysosomes can break down contents that have been absorbed into the cell, right, by invaginating the plasma membrane, taking in different types of microorganisms, different types of solutes. Okay, so different types of structures here, sucked into the cell essentially via what's called endocytosis. Okay, this forms a little vesicle called an endosome. Okay, and the lysosome, a vesicle from the Golgi apparatus filled with proteins that we just transcribed, translated, processed, and packed into this little vesicle here. This lysosome can then fuse with the endosome essentially allowing all of these substances here to be digested. And just one final note, this process of taking in substances from outside the cell is called endocytosis. There are a couple different types of endocytosis. Phago or phagocytosis is essentially taking really big things in. Penocytosis is essentially like the drinking form if this is like the eating form. So this is taking in fluids and smaller solutes. And finally, sometimes this process is mediated by a receptor, which of course was put there through this process as well. Okay, so thank you so much for watching. This, again, has been the process of protein synthesis: transcription, translation, post-translational processing, and ultimately the fate of these different proteins as digestive enzymes within lysosomes, insertion of membrane proteins, and exocytosis or secretion of different solutes outside the cell. Thank you so much for watching.